कोई नहीं, आज कम हो गया क्या? नहीं नहीं, आपके इसमें चालू कर दो, उसमें स्टार्ट कर दो आपके मोबाइल में. We'll wait if someone is joining. May I know who has joined? If you could comment your name. Hi. Yes. एक तो दो ये. Yeah. We'll just wait for 1 or 2 minutes and then we'll start. And I'm a bit tired today. I had almost 5 lectures up till now, and I have more to be conducted. So very brief overview, only 5-10 questions here and there, only 10-15 minutes around there. So just stay tuned with me for 2 minutes more and then we'll start. Just 2 minutes. In the meantime, you can comment who is present there. A very good afternoon. At least you can comment. I'll come to know who has joined. So you don't want to reveal who has joined. Anyways, just a moment. In 1 minute, we'll start. I'll just get my water bottle. Good afternoon. Raj. Good afternoon. Raj, you are here for what? You're now in 3rd year, Raj? Yes, Prachi. I don't know whether the birthday boy has joined. Vivek has joined. Today is Vivek's birthday. You can wish him happy birthday in the meantime. Let's go. I think let's not wait much now. Good afternoon. Yes, Vivek. Okay, there you go. As I told, a very simple one. It will hardly take 15-20 minutes. By the time people will join, our session would have been over. Anyway, so yes, here you are, the first one to go without wasting much time. ATP is present in the cell complexed with, like you have a lot of ATP present there. ATP is being formed, ATP is being used. Inside the cell, it is complexed with a particular ion. Right? Can you tell it is complexed with what? It is complexed with iron, zinc, magnesium, or copper. Yes, anyone out there? ATP inside the cell is present as a complex of. ATP is not always alone. ATP is always bound to something. So you know what ATP is bound to. Just let me clean this lens for you all so you can see more clearer. Yes, great. Vivek says it is magnesium. Yes, Manasvi from Pune, it is magnesium. Yeah, you're right. ATP in the cell is present as a complex of magnesium. Let's go to the next one. GTP plays a major role in, GTP is important in what? In protein synthesis, in nucleic acid synthesis, in carbohydrate synthesis, or in lipid synthesis. The immediate source of energy is GTP in one of these. Can you tell where? Yes, Prachi, it is magnesium, right? Now here GTP, I am saying GTP. GTP serves as a source of energy for which particular process? You can comment in the comment box as A, B, C or D. Protein, nucleic acid, carbohydrate or your lipid synthesis. Yes, GTP. GTP, remember, is a donor of energy for the phenomenon of protein synthesis. Remember, GTP is important in protein synthesis. So remember for your exam. And pardon me because my energy is low today. I'm a lot of tired today. I don't know. Mala sote ka tumhala pan hote hai. I don't know. Anyways, no. Priyanka, it's not for carbohydrates. GTP acts as a source of energy for protein synthesis. Pentameric antibody which has five what you call as units together, right? Generally, when you draw this antibody, you draw it like this. You have five what you call as antibodies which are joined together, 1, 2, 3, 4 and 5. So which is this pentameric antibody? It is M, it is E, it is G or it is D. What do you think? Which is a pentameric antibody? I also told that is the same antibody which comes first when antigen enters your body. Great. It is IgM having a valency of approximately 10. You know that they are joined together by a particular chain. Whenever an antibody is oligomeric, having many what you call as antibodies coming together, many antibody subunits, basic units coming together, they are connected by a particular chain. Can you tell which is that chain? It is A chain, it is J chain, it is S chain or it is V chain. Which is that chain which joins all of these? कौन join करता है इन सबको, ये पांचों को? Which chain joins it? Yes, please. Yes, great. It is J chain, right? IgA is missing here, right? Your secretions, they generally contain IgA. From mother to child also, from colostrum also, this IgA, it flows. Anyways, AA, BB, CC, DDE. Here what you can see. I hope you can really see it. So this is the heterozygous here, this is heterozygous here and heterozygous here. Heterozygous at B, heterozygous at C, heterozygous at D, right? How many types of gametes it will form now? What do you really think? Just yesterday we did this, right? You have to just use that formula, 2 raised to heterozygotes. So here heterozygotes are how many? 3. So heterozygotes are 3, so it is 2 raised to 3. So 2 raised to 3 is how much? 2 raised to 3. एवढा mathematics आलं पाहिजे ना, please come on, please be quick. I want to just like wind up this and meet you tomorrow again. It is not 16. 2 raised to 3, please someone do it. 2 2s are 4, 4 2s are I don't know. Yes, B. Yes, Pramod, it is B. That is your 8th grade. So it is your 8th grade. Great. Pramod kas काय join झालाय? Anyways, great Pramod. Archaebacteria were separated by sequencing. Like when we got separated your eubacteria and archaebacteria from your eukaryotes, like who did this? I suppose Carl Woos did this and added the 3 domains of life, right? So in addition of these 3 domains of life, who was playing a major role? The 16S rRNA, the 23S rRNA, EFTU, or your genome, right? What do you think like was playing a major role? What was the thing which brought a revolution? Yes, please, I'm here only. Yes, 16S rRNA, right? One of the what you call as your molecular clocks. Citric acid cycle does not directly involve the citric acid cycle, the TCA cycle, the Krebs cycle, whatever you call it, part of aerobic respiration taking place in the matrix of the mitochondria. It does not involve what? Molecular oxygen, succinate, FED or NAD. What does it does not involve directly? Yes, Raj, it is 16S RNA, great. I'm looking at your chats. I'm not looking at the answers. I'm taking only those questions whose answers I know. She says B, succinate. No, succinate astagi. You have succinate in the citric acid cycle, right? You have OA combined with acetyl CoA giving rise to your citrate, isocitrate, then your alpha ketoglutarate, then succinyl CoA, then succinate. Succinate is formed. Succinate astana. Who said succinate? Succinate is there. It is a part of citric acid cycle. Yeah, Vivek says A. Great, great, great. Molecular oxygen is not directly what you call as the part of citric acid cycle. Molecular oxygen comes into picture at the end. वो हीरो कैसे लास्ट में आता वैसे ये ऑक्सीजन भी लास्ट में आता during the third step, major step of aerobic respiration, which you call as oxidative phosphorylation or electron transport chain. Can you tell me that in mitochondria, where does this ETS, electron transport system, take place? A, matrix, B, outer membrane, C, inner membrane, D, I don't know. So you can just comment, like if you say it is matrix, you can comment M. If it is outer membrane, you think OM. If it is inner membrane, IM. And I don't know, you can have a smiley there. राज क्रेप्स सायकल नाही विचारली. मी विचारले ETS कुठं होतं? Where does ETS takes place? ओहो विवेक, मॅट्रिक्स मध्ये ETS होत नाही. यू आर सो ग्लॅड, रादर आय एम सो ग्लॅड की तुम्ही माझ्या समोर नाही येत, नाही तर खाल्ली असती झापड. Oh, yes, yes. Sorry. Vivek has said IM. It is inner membrane. Great. Sorry. Oh, I'm so sorry, Vivek. It is inner membrane. So, yes. So, ETS takes place in the inner membrane. Yes. Sorry. Next one. Cell not having a well-defined nucleus may contain. A cell which is not having a well-defined nucleus, but it may still contain a Golgi complex, a mitochondria, a ribosome, or all of these. You can comment A, B, C, D. The cell which is not having a very defined nucleus, a prokaryotic cell, I suppose, a bacterial cell. So bacterial cell will contain. What will this bacterial cell contain? Golgi complex, mitochondria, ribosome, or all of these? Manaswi says all of these. No, a prokaryotic cell doesn't contain all of these. What does a prokaryotic cell contain out of all these? Priyanka says C. Again, Raj says D. Raj, it's not D. Prokaryotic cell doesn't contain all of these. Priyanka says C. Ribosome. And I think ribosome should be a right answer. Yes, ribosomes are the ones which are also present in prokaryotes. Can you tell me ribosomes in prokaryotes are of which type? 70S or 80S? No, it's not B. It's ribosome. We have got the answer. It's ribosome. So they are 70S or 80S. Now the question is, the ribosomes in prokaryotes are 70S or 80S? 70S. Great. And S stands for Swedberg's unit. Anyways, an increase in the pK of an acid an increase, I hope you can see the screen. Yeah, I can also see the screen now. Increase in the pK of an acid makes an acid. Like what happens to this pKa of an acid if it increases? If pK increases, the acid becomes stronger or it becomes weaker. What do you think? Yes, pKa बढ़ गया. pKa is what? How do you define pKa? pKa is negative log of Ka, or you can say pKa is 1 upon like log of Ka. So if pK, I say it increases, the acid will become weaker or stronger. If pK increases, right, the Ka decreases, and Ka is what? Dissociation constant. So dissociation of the acid, it decreases. So if dissociation of acid it decreases, it is going to produce less H plus. If it is going to produce less, if it is going to produce less H plus, it will be more stronger or weaker. So pKa increase, Ka decrease, dissociation decrease. So acid becomes, Manasvi says, weaker. Others? Dissociation कमी झालं. Yes, Manasvi. So therefore it becomes A, that is weaker. Dissociation कमी झालं, H plus will be produced less, and therefore the acid will become weak. So it's A. Go to the next one. Ratio of specific activity to the purified extract to that of the crude extract. So when you are purifying the enzyme, probably they are talking about. So the ratio of the specific activity of the purified extract to the crude extract. Purified extract का इतना specific activity था, upon crude extract का इतना specific activity था. वो जो अगर आप ratio लेते of purified extract and the crude extract, तो वो क्या हो जाता है? Like it gives you the yield, it gives you the units, it gives you units per mg, or it gives you the purification factor. What do you think? Yes, please. Manasvi says B, purification factor. Great, great. You're going great, Manasvi. It gives you the purification factor, which you mention as folds, purification folds. We say that enzyme got so many times purified, twice purified than that, 4 times purified than that, 10 times purified than that, right? So that purification factor, it is the ratio of your specific activity of your purified extract and crude extract. Can you tell me, like, then specific activity is defined as what? Specific activity of the enzyme, it's units of enzyme upon what? Can you tell me? Specific activity is units of enzyme upon what? B. Prachi, it is B. This is B, purification factor, the 9th one. I'm now talking specific activity is units of enzyme upon dash. When you are purifying the enzyme, you know that you're removing the impurities, so you have to calculate and monitor that time to time. So specific activity, you have to calculate the specific activity is units of enzyme upon dash. No, it's not enzyme activity. This units of enzyme is enzyme activity actually. So it is activity of the enzyme upon what? I suggest you to go in the same channel and watch the video on purification of the enzymes, or you can just ping me on 9823683317, and I'll share you the video of all the what you call as terminologies which are involved in purification of the enzyme and how they are calculated with the entire examples in that you will get. So you can ping me here, I'll share you the video there. Concentration, but Prachi, can you tell me what? Concentration of what? Concentration of enzyme, concentration of carbohydrates, concentration of protein, concentration of what? Prachi is right. It is the units of enzyme upon concentration. But I want to know concentration of what? Prathamesh has not joined today. Prathamesh, Vivek has joined, I suppose. Ross has not joined, I think, today. Patil, Prasanna has not joined. Many have not joined. Those who have joined now, Prachi, Priyanka, Vivek, please tell me. It is concentration of what? It is concentration of protein. It is concentration of protein. Great, great. Yes, Prachi. Yes, Vivek. It is concentration of protein. Okay. Next one. Only one type of monomer will be released by hydrolysis of. अगर आप हाइड्रोलिसिस करते हैं इन सब चीजों का तो एक ही टाइप का मोनोमर आएगा. किसमें? DNA, RNA, starch or triacylglycerol. If you break them open, you will get only one type of monomer. You will not get any other type of monomer. After cleaving DNA, after cleaving RNA, after cleaving starch or after cleaving triacylglycerol. किसको कट करने के बाद आपको एक ही टाइप के मोनोमेरिक यूनिट्स मिलेंगे. Only one type. Yes, please. You cut DNA, you will get all the kinds of stuffs, the different types of nucleotides you will get here also. You will get different types of nucleotides. Triacylglycerol, you will get fatty acids, you will get glycerol. Starch, if you cleave starch, what you will get? Yes, Priyanka, it is starch. I want to know when you cleave starch, what you will get. Starch को degrade कर करके final monomeric unit आपको क्या मिलता है? Yes, Raj, what do you get after cleaving starch? Starch को हम degrade करते जाएंगे तो आपको क्या मिलेगा छोटे छोटे units? Great. Raj says it is glucose. Great. Yesterday only we told. Intracellular parasite. Which of these is an intracellular parasite? The one which is living inside the cell. It is Streptococcus, E. coli, Clostridium or Rickettsia. Which of them lives inside the cell? Yes, please. Streptococcus, E. coli, Clostridium or Rickettsia. Which of these lives inside the cell? इंट्रासेल्युलर पैरासाइट जो सेल के अंदर है भाई, द वन विच इज लिविंग इनसाइड द सेल. स्ट्रेप्टोकोकस, ई कोलाई, क्लॉस्ट्रीडियम और रिकेट्शिया. हु लिव्स इनसाइड द सेल? You can comment as A, B, C, D also. Streptococcus, E. coli, Clostridium and Rickettsia. Google तो नहीं करते ना तुम? Oh, it's simple. Yeah. No, it's not E. coli. Anyone else? कुछ तो बोल दो. It is D. Rickettsia. Right. So rickettsial parasites, right? They are what you call as growing inside the host cell. You have Plasmodium, the one which causes malaria. So Plasmodium, it belongs to a ciliate protozoan, a flagellate protozoan, amoeboid protozoan or a sporozoan. No, E. coli is not inside the cell, beta. E. coli is outside the cell. Cell के अंदर जाने वाला कौन है? Rickettsia. You know that rickettsial fever, the Q fever, the all that things, Rocky Mountain spotted fever and all that stuff related to chlamydia, rickettsia. Anyways, बाद में देखेंगे कभी वो. Plasmodium belongs to what? Yes, D. She says sporozoa. Great. So they are sporozoa. They are non-motile. The last one you have, the purine is replaced by another purine. So you have one purine replaced by another purine. What it is? Transition, frame shift, transversion or silent mutation. One purine. आपने एक ए के जगह पे एक जी लगा दिया, या एक जी के जगह पे एक ए लगा दिया, या टी के जगह पे सी लगा दिया है, सी के जगह पे टी लगा दिया. So this particular mutation is what? Transition, transversion, frame shift or silent mutation? Transition. Great, Vivek. It is what is called as A, that is transition. Okay. So as I said that today I am a bit tired. Actually, I had a lot of lectures today and a lot of stuff going on. So today probably will be ending here. But you all are having your exams on coming Sunday, right? On Sunday, it's at what time approximately, right? At different centers you are having your exams. So it's at what time approximately? It is what is the reporting time at the center on Sunday? 11 to 1 you are having the exam, right? 10 a.m. Okay. So what we can do is, okay, 10:30, something around like that. Okay, great. Prachi is still doing MCQs. Anyways, what I wanted to ask is कि कल सुबह 7 बजे जमेगा आपको? I'm not taking it at night probably. क्या 9 बजे 9:30 बजे कल जैसे चल सकता है? थोड़ा 10 तो क्वेश्चन लेते हम. Late evening 9:30 can work with you all? क्योंकि आज और कल सुबह का ही दिन है बस. दैट्स इट. देन यू आर डन. यू वांट मी टू कंटिन्यू आफ्टर दैट आल्सो? उसके बाद देखेंगे. On संध्याकाळी in the evening, how many of you can join at 9:30 if I am taking? Vivek, you can join. Today is your birthday. Okay, so if 9:30 is fine, then it's fine for me also. Okay, I'll drop in a message if I'm taking at 9:30. So we'll meet today at 9:30. Tomorrow I have a lot of classes. Tomorrow around 7 to 8 lectures. So I'm very doubtful whether I'll be able to take a session tomorrow. But let's see. जमत असेल तर नक्की घेऊयात. And काही ना काही वाचत राहा. And all the best. Bye bye. Take care. बाय बाय तर म्हणा मला, म्हणजे I'll feel good. Bye bye. Take care. Bye bye Raj. Bye Prachi. Bye.